Our next speaker is John Tazare from the London School of Hygiene and Tropical Medicine, where he works in the Medical Statistics Department as a statistical pharmacoepidemiologist. His research is around the use and development of causal inference methods for investigating effectiveness and safety of medications from routinely collected EHR data. So he's definitely in the right place. And John, we look forward to your talk about COVID therapeutics.
Thanks very much. Thanks, Jonathan. So yeah, in February 2023, NICE updated their treatment guidance for COVID-19 therapeutics and sort of cited non-randomized evidence consideration from OpenSAFELY. And so in this talk, I'm going to kind of walk through how we got to this position. So I'm going to give a short background on the rollout of these medications and describe our process curating this new data set. I'm going to give a brief summary of some of the effectiveness research we've done on this, and then wrap up with some opportunities for future research, as well as some of the impact of this work. So in December 2021, CMDUs was launched across England to provide medications in community settings treating symptomatic COVID-19 patients at high risk of severe outcomes. And in this early period, neutralizing monoclonal antibodies and antivirals were rolled out, and the two most frequently prescribed medications were sotrovimab and molnupiravir. And the approval for these medications was largely based on evidence from two phase 3 randomized control trials, the COMET ICE trial, which looked at found a 79% relative risk reduction for all-cause mortality for all-cause hospitalization and death, comparing sotrovimab versus placebo, and the MOVE-OUT trial, which found a 30% reduction comparing molnupiravir against placebo. So this opened up opportunities for observational data to contribute to the rollout of these medications, and this was particularly important given key differences between trials and observational data. So firstly, the trials were conducted in unvaccinated populations, and at this time in the UK, vaccination levels were now quite high. And it was also important in terms of rapidly emerging variants, where the trials were conducted prior to the Omicron variant. So we identified these three research aims: rollout monitoring, so how the medications were being rolled out and to whom, comparative effectiveness of sotrovimab versus molnupiravir, and also effectiveness and the potential to do non-user comparisons. But first, to do all of this, we needed the data. And we first received the COVID-19 therapeutics data set on the 27th of January 2022, and we received weekly updates thereafter. This was supplied by NHS England via the BlueTech system, and we obtained all information on approved medications in inpatient and outpatient settings, and we were able to link this to primary care records securely in line with the OpenSAFELY practices in the data warehouse of TPP. And the OpenSAFELY team were able to sort of generate and sort of create sort of queryable data fields for us within a month. And here just at the bottom, I've included a little snippet of the Python code we kind of use to define and identify the patients who are receiving these treatments. So our first step was really around data curation and a careful and systematic data quality and content assessment. This included reviewing data schema, data formats, completeness, and data values. And the key fields that we were going to go on to use for the research included inpatient and outpatient information, which treatment people received, and the treatment date that they received. And this was very well completed data. Other fields were less complete, so surrounding the region, particular high-risk groups, and symptom onset. And then this is all sort of being summarized in a data report that will be published soon if you'd like more information. That's at the bottom. So our second step was really to then, having brought the data in, was to start monitoring. So we linked the COVID-19 therapeutics data set to primary care, secondary care, mortality, and COVID-19 testing data. And from this, we were then able to sort of define and identify the potentially eligible patients for treatment. And this was based on being a member of a high-risk group and confirmation of COVID-19 infection defined either through a positive PCR or lateral flow test. And looking at this sort of population over time, this allowed us to look at coverage variation between key clinical, regional, and population subgroups. This is work led and published by Amelia Green. But I also just want to highlight the role in which this work, before it was a publication, was rolled out as a weekly report, as you can see up on the screen. And the website that we sort of posted that is there. And again, this was done within 2 months of the data being first received. And on the right-hand side, you can just get a flavour of the sort of things that we were including in that. And this shows the treatments received over time. And this was really invaluable for us because, as we were saying earlier, we don't get to see the data ourselves when we're working with OpenSAFELY, and these sorts of reports were really key in terms of both understanding the feasibility of potential analyses later on, but also getting to understand how to define key concepts. So, for example, around the high-risk criteria. And the second thing was, at this point, we've now built up a large amount of programming code, which we can then reuse efficiently going forward into the effectiveness research. So now we know that which of these medications were being used by people. Our first question was around the comparative effectiveness of these treatments, and in particular, sotrovimab versus molnupiravir, and this is work led by Bang Zhen from the London School of Hygiene. So the first aim was just to estimate comparative effectiveness of sotrovimab versus molnupiravir on preventing severe outcomes in these non-hospitalized high-risk patients. So we identified all patients with non-hospitalized treatment records for either of these medications initially between December 16th and February 10th. And then we compared the following treatment strategies to either initiate treatment with sotrovimab or molnupiravir. And the primary outcome was COVID-19 related hospitalization or death based on the ICD-10 codes presented on the slide. So for the follow-up, individuals were followed from treatment initiation until the earliest of having an outcome, 28 days since treatment, initiation of a second MAB or antiviral, death, patient deregistration, or the study end. And we performed a propensity score analysis, estimating propensity scores using logistic regression, adjusted for baseline covariates, and then estimated 28-day hazard ratios were obtained from a weighted Cox model. So the results were as follows. We identified about 3,300 sotrovimab users and 2,700 molnupiravir users. And a summary of the characteristics of those two groups is presented in the table on the left. And we can see that they were broadly similar, with the sotrovimab users being slightly younger, having slightly a lower burden of immunization and a higher burden of renal disease. We observed 32 outcome events for the sotrovimab group and 55 for the molnupiravir group. And again, we can see there's a higher proportion of COVID-19 deaths among the molnupiravir group. So this forest plot presents the results for the primary analysis, and we can basically see that that sotrovimab was sort of consistently associated with a lower hazard of COVID-19 related outcomes compared to the molnupiravir group. And reassuringly, this was sort of fairly constant across a sort of wide range of supplementary analyses as well. So what were our sort of conclusions from this comparative effectiveness research? So really, unmeasured confounding we thought was sort of likely to be limited because of the use of this ACNU design. And importantly, this initial study anyway was during a period where there was true sort of clinical equipoise between the treatments. They were both first line at the time, and it was reassuring to obtain results that were broadly consistent when triangulating our results against those from trials and in vitro and in vivo studies. And again, this sort of framework was sort of developed and reused very efficiently to then look at subsequent analyses, looking at Paxlovid versus sotrovimab and effects across different eras of Omicron and beyond, and as well as looking at patients with kidney replacement therapy for whom there was a key contraindication later on for one of the key drugs of interest. So we saw that there was evidence then of sotrovimab perhaps performing better than molnupiravir, but we were also interested in if we could actually perform these non-user comparisons. So I'm going to sort of go through and summarize some of that work in the next few slides, and that was work led by myself and Linda Nabb from Oxford. So the aim of this work was to estimate the hazard of COVID-19 related hospitalization or death, so the same outcome as before within 28 days associated with the use of either sotrovimab or molnupiravir versus no treatment in these non-hospitalized high-risk patients. So we could imagine a hypothetical trial where people are randomized to treatment or not at the time of positive COVID-19 test, but in practice this wasn't quite compatible with the treatment guidance at the time, which was to recommend treatment within 5 days of a positive test. And this posed sort of key challenges for this sort of research. So firstly, confounding. Assignment was hypothesized to be largely driven based upon symptoms at the time. Availability of data, baseline or post-baseline data on severity and disease was either unavailable or fairly incomplete. And it was also important in terms of avoiding immortal time. So people receiving treatment at day 4 must have survived until day 4. So we sort of set about using the target trial framework to sort of systematically try and characterize and where possible alleviate some of these concerns. So in the next few slides, I'm going to briefly give you an overview of that target trial specification. I'm not going to cover all of the eligibility criteria, but just to say that the sort of base cohort we're working with here are a group of patients who have got symptomatic COVID infection based on either a positive test and they're a member of a high-risk group. So we then were interested in comparing 2 treatment strategies where patients were randomized to, on the day of diagnosis, to either initiate treatment within 5 days or not initiate treatment within 5 days. And this then sort of led to us sort of emulating 3 trials considering treatment as either sotrovimab or molnupiravir, molnupiravir only, and sotrovimab only. So to emulate the treatment assignment component of the target trial specification, we used a clone, censor, and weighting approach. So here patients were copied or cloned, and one clone was assigned to each of the treatment strategies. When patients deviated from that assigned treatment strategy, those clones were censored, and we used inverse probability of censoring weights to adjust for the induced selection bias. As before, this outcome was COVID-19 related hospitalization, admission, or death. And the follow-up, so index date in this case was the date of diagnosis, so the date of positive test, and individuals were followed up until the first of either 28 days, the occurrence of an outcome, administrative censoring, or non-COVID related death. So the causal contrast of interest was a sort of observational analog to a per protocol effect where we are accounting for treatment strategy deviations between index date and day 4, but not beyond this. And we summarized this using 28-day hazard ratios and difference in 28-day survival. So this slide shows the results from the primary analysis. So overall, we did not find an effect when comparing treated with any of these 2 drugs versus untreated. And similarly, we didn't find an effect of molnupiravir versus untreated. Sotrovimab did appear to be associated with a low hazard of COVID-19 outcomes versus untreated. And we can sort of see from the estimated the weighted cumulative Kaplan-Meier estimates on this slide that we saw a very similar pattern of results here too. So the sort of conclusions from this effectiveness research was that, well, the target trial framework helped to identify and characterize key challenges in estimating effectiveness. Some of these challenges could be fixed using appropriate design and methods. And for example, particularly relevant here was the use of clone sensor weighting to avoid immortal time. However, we still had sort of concerns, and in particular residual confounding, especially due to missing symptomatic data, likely remained. In the case of baseline information, we were able to investigate this using bias-adjusted quantitative bias analysis. And for this, we used parameters that were directly informed by CMDU service evaluation data. However, there was no data on post-baseline progression or severity, and it just wasn't possible to account for this in any of our analyses. Finally, I just want to highlight that there's sort of ongoing work that's taking a slightly different approach using sequential trial designs to look at Paxlovid research. And I just want to finish in the next couple of slides by sort of giving kind of a summary of some of the strengths and sort of impact of this initial bit of work, and then highlight some possible opportunities for using this kind of framework in the future. So I think the first thing really is that that sort of drove a lot of the success of this work was being able to rapidly and early on after receiving this data, link it and make it operationalizable for research. And that was really fundamental to what came next. I then think that sort of by combining both near real-time data and robust causal inference approaches, we were able to sort of generate evidence, fairly reliable and robustable evidence that contributed to living treatment guidance in the UK. And these results also went on to inform international guidelines in Canada, France, Germany, and Japan. And I think, as I said, the key sort of takeaway really from this talk is that the framework that has been presented is really adaptable, and using OpenSAFELY, we can apply and investigate effectiveness and safety of potentially any drug of interest. So how do we build on this going forward? So I think firstly, data availability, and we're working really hard to try and improve our ability to embed and link novel data sets routinely. And also in terms of validating analytical pipelines. So we're hopefully working collaboratively to try and sort of understand and implement the best practice application of key causal analysis strategies, which may help us answer these sorts of questions. I just want to acknowledge the core COVID-19 treatment working group that's listed here on the slide and our funding, and thanks very much everyone for listening.